Good day, students. I welcome you to study session 3 of NSC 206, which will discuss amino acid and the chemistry of protein. Proteins are natural polymer molecules consisting of amino acid units. Despite the wide diversity of proteins, all proteins are composed of long chains of smaller building block, which is known as the amino acid. In the previous study session, you have been introduced to the chemistry of carbohydrates, and proteins, being one of the most important classes of biomolecules, serve as the basis for the major structural components of animal and human tissue. At the end of this study session, you should be able to describe amino acids. You should be able to define the term protein. You should be able to explain different classes of protein and the structural levels of protein. First, let's look at amino acids. Amino acids are important organic compounds composed of amine, which is the NH2, like you can see on your screen, the amino group. And it also contains the carboxyl acid group, which is the COOH group, along with a side chain specific to each amino acid. The key elements of an amino acid are carbon, hydrogen, oxygen, and nitrogen, though other elements are found in the side chains of certain amino acids. The amino acids are colorless, non-volatile, crystalline solid, melting and decomposing at temperatures above 200 degrees centigrade. These melting temperatures are more like those of inorganic salts than those of amines or organic acids, and they indicate that the structures of the amino acids in the solid state and in neutral solution are best represented as having both a negatively charged carboxyl group and positively charged amino group. What is protein? Proteins are most important constituents of cell membranes and the cytoplasm. The muscles and blood plasma also contain certain specific proteins. Proteins are natural substances with high molecular weight ranging from 5,000 to many millions. Besides carbon, hydrogen, and oxygen, they also contain nitrogen and sometimes sulfur and phosphorus. Proteins are natural polymer molecules consisting of amino acid units. The number of amino acids in protein may range from 2 to several thousand. Protein-containing foods are essential for living organisms because protein is the most important biological molecules in building up and maintaining the structure of the body, giving as much energy as carbohydrates in the course of metabolism in the body. What are the classification of protein? Though there's no universally acceptable classification system, proteins may be classified on the basis of their composition. It can be classified on the basis of solubility, shape, and on their biological functions. The two compositional classifications of protein are simple protein and conjugate protein. The simple protein yields only amino acids and no other major organic or inorganic hydrolysis products, while conjugated proteins yield amino acid and other organic and inorganic components. Classification of protein based on solubility. We can have albumins, and these are proteins such as egg albumin and serum albumin, which are readily soluble in water and coagulated by heat. We can also have globulins, and these proteins are present in serum, muscle, and other tissues and are soluble in dilute salt solution but sparingly in water. We can have histones too. Histones are present in glandular tissues, thymus, like the thymus and the pancreas, and is soluble in water, and they combine with nucleic acid in the cells, and on hydrolysis they yield basic amino acids. The protein structure. There are three-dimensional geometry of a protein molecule, which is so important to its function. There are 4 levels of organizational structure are used to describe protein. We have the primary structure, we have the secondary structure, we have the tertiary structure, and we have the quaternary structure. Let's look at the primary structure of protein. The primary structure of protein refers to the linear numbers and order of the amino acids present. Protein contains between 50 and 2,000 amino acid residues. The convention for the designation of the order of amino acid is that the end terminal end, that is the end bearing the residue with the free amino acid group, is to the left. And the number one amino acid, the carboxyl terminal end, that is the end with the residue containing the free carboxyl group, is to the right. Looking at the secondary structure of protein, it is usually referred to as the coiling or folding of a polypeptide chain that gives the protein its 3D shape. As you can see on the picture on your screen, the coiling shape. There are two types of the secondary structures observed in protein. We can have the alpha helix structure. This structure, which resembles a coiled spring and is secured by hydrogen bonding in the polypeptide chain. We can also have the beta pleated sheet, which appears to be folded or pleated and is held together by the hydrogen bond between polypeptide units of the folded chain that lie adjacent to one another. We can also have the tertiary structure of protein, which is the comprehensive 3D structure of polypeptide chain of a protein. There are several types of bonds and forces that hold a protein in its tertiary structure. Hydrophobic interactions greatly contribute to the folding and shaping of a protein. The R group of the amino acid is either hydrophobic or hydrophilic. The amino acids with hydrophilic R groups will seek contact with their aqueous environment, while the amino acid with hydrophobic R groups will seek to avoid water and position themselves towards the center of the protein. The quaternary structure of protein is the structure of a protein macromolecule that are formed by interactions between multiple polypeptide chains. Each polypeptide chain is referred to as a subunit. The proteins with quaternary structure may consist of more than one of the same type of protein subunits. They may also be comprised of different subunits. Hemoglobin is an example of a protein with this quaternary structure. Hemoglobin, which is found in the blood, is an iron-containing protein and it binds oxygen molecules. It contains about 4 subunits, of which it has 2 alpha subunits and 2 beta subunits. Thank you for listening. Bye for now.